The overall objective of our module is really to enable the efficient and effective application of genomic technology in breeding and mainstreaming that within the AFS or CRP network. So again, for those of you unfamiliar with CRP language, AFS is just Agri-Food Systems. It's a kind of alternate name as far as I understand it. For what we have referred to as the CRP networks, and then accelerating the rate of genetic gain delivered in farmers' fields. And this module is, I think it's been pointed out a couple times, is a service module. We're really here to help make breeding more efficient, however that looks. We want to assist with the assessment of if and where genotyping is appropriate, and then search for lowest cost solutions wherever there are kind of broad applications required by the wide community of AFS teams. And I think kind of touching on what George was sharing a little bit earlier, this idea of having too much choice is a bit of a problem. And so one of the things we're identifying is that, we want to kind of distill that down to what are the most common needs and find the best possible solutions, understanding that finding a solution for a broad community may not be perfect for everyone, but I think what you'll see is that the kind of guiding principle behind what we're doing is that wherever we can kind of aggregate our demand for a particular service, it gives us better position to negotiate for price. So we don't see ourselves as finding many solutions for many people. It's more about what are the big needs, and then trying to find good answers in those spaces, and then assisting with efficient implementation as needed, and wherever we're able to provide some advice in that space. So these are the three main applications that we're focused on right now. Genetic QC seems to be kind of an underlying focus area across all of the species. Haplotype enrichment we talk about, that's sort of, you can think of that as marker-assisted selection or forward breeding. That's at the core of what the HTBG project was, and we'll continue to work on that model. And then, of course, looking for solutions to support wider application of genomic selection through our breeding programs. One of the key things that we're looking at doing together with Module 2 is really cost-benefit analysis. We want to do this for MAS and GS workflows within the context of the teams and networks. Just to kind of back up a little bit, the terms I'll use here, teams and networks, I think of more as a team. Typically you have, and we're still working out some of the maybe vernacular that we use for defining different terms. But if you think of an individual scientist with their staff, we would kind of think of that as a breeding program, and then a group of scientists within an institution working on the same crop or maybe towards the same product profile would be more of a team. And then a series of institutions working together on a similar crop variety replacement strategy would be more of a network. And I like to use the term network instead of community of practice, because for me, community of practice doesn't really infer any accountability. We can be a part of a community of practice and share ideas and go home and nothing really, we don't have any accountability to one another. Whereas the network, in the breeding space, a network implies that, I'm sending you plots, you're sending me plots, we're returning each other data. We're working together to try and we're all successful together. We don't work independently. Okay, so our milestone here is to have all member breeding teams have implemented cost-benefit analysis for genotypic data supported workflows. That's a bit ambitious, but we hope over the life cycle of the 4 or 5 years, we hope to be able to be doing this kind of routinely with all of our breeding teams. And then again, back to stage gate decisions, we move forward with green light applications with bring value, and we either abort or further improve red light applications. So this is again, kind of, I think George touched on it, not really focused on the tool driving the strategy. It's more about, does this tool make sense in the context of what you're trying to do? And if it does, yeah, then we go forward and, we help try to figure out the best strategy. And if it doesn't, then, we either go back to the drawing board or use a different tool. Okay, so I wanted to talk just a little bit about program optimization and thinking about this in terms of, what you might consider, sort of an evolutionary space of ideas, and you have some people trying new ideas and kind of moving towards more optimization. And in this particular example, you could think about, on this axis, maybe we've got genetic gain, some measure of genetic gain, and, and our breeding teams are on one of these hills in some way, trying to think about, okay, there are new opportunities, and, the field of plant breeding is evolving. How do we start exploring some other opportunities when in fact it might, we might have to go down the hill a little bit to get over to a higher peak opportunity. So in this case, you can think about, in some ways, maybe we're, kind of in this space breeding, and we're optimizing our programs through a number of things, and then suddenly, as we're seeing in the last 10 years, the cost of genotyping using sequence-based information is coming down pretty dramatically. So there may be opportunities to improve our efficiency if we can effectively utilize that information, if we can get ourselves from this peak, onto this hill. And what we recognize is that, in fact, when you do proof of concept experiments, typically you're kind of going backwards to go forwards a little bit, right? And historically, we've done quite a bit of proof of concept. In fact, we did a large proof of concept or proof of principle type experiment at CIMMIT last year, where it involved a lot of moving pieces, and it was very complicated to try and employ. And we recognized that overall, it was probably fairly inefficient and involved a lot of different people. But we hope to be able to learn from that, and then to be able to develop a pipeline to be able to do those same steps much more efficiently, so that other teams trying to implement similar ideas would be able to use what we learned from here to move from this, say in this example, from this peak over onto this hill and start climbing more efficiently in the future. Part of Module 3 will be focused on training, looking at use cases and implementation guidelines documented in the EIB toolbox, and we're actually sourcing those from the CG centers anywhere anyone's actually got a good practice that they're already implementing or examples of things they're doing. These are sort of best practices as we see. And then part of this week's gathering, we've got folks from the Gobi team who will be doing training, the IGSS, who you'll meet here, Clay Sneller will be joining us for the rest of the workshop. So we want to be able to help provide those opportunities for breeding programs who may want to learn in terms of being able to effectively implement strategies. So one of our milestones is a year-on-year increase in the application of workflows within the breeding networks. Again, those are reviewed before we do that, but assuming that some of these workflows make sense economically for your breeding program, then we'd like to see them implemented. And then document refinement and improvement of strategies over time. Okay, in the area of cost reduction, just to give kind of a high-level picture of what we're trying to do, again, back to that idea of aggregated demand, we want to be able to negotiate with vendors. It's quite similar, I think. George's analogy is helpful for me as well, because again, you go out into this space and there's at least 5 or 6 different genotyping concepts or ways you could do it, and each have pros and cons. And so among the community of breeder or molecular breeding teams that exist within the CGIR, there are constantly vendors talking with each of us about, well, here's the next greatest thing, and how do we sort of collectively pull that information together, funnel it to one person to help us sort of strategically make decisions about what makes sense for the future of where we're going together. And we, admittedly, not that we'll have the best solution all the time, and some individual teams may be from time to time making decisions outside of this framework, but the idea here is to try and ultimately aggregate demand to negotiate for price, and then we'd like to coordinate the technology prospecting part so that we can share information about these platforms across the CG Center, so that we're not all doing parallel proof of concept experiments with Illumina this week and some other service provider next week, and That's the idea here. And then a high-level goal for us is to really negotiate contracts with service providers, and these are sort of our initial metrics. We want to try to reach $1.50 per sample SNP genotyping. This is the HTPG aspiration from the outset, and if we meet our aggregated demand targets, this is more or less where we'll get to. And then we're really now just focusing on finding good solutions in the space that will enable teams to do genomic selection. So this again is kind of our target. It's a bit of a moving target, but we think we'd like to see within the 4 to 5 year time frame, you know, a lower than $8 per sample cost for DNA extraction and adequate markers to do GS together with bioinformatics, so that it's pretty much ready to go for a breeding team. Yeah, so we also are thinking about logistics support to enable us to scale across multiple crops, and so part of the platform, the module will have a person focused on logistics support, and our milestones here are to see somewhere in the neighborhood of 800,000 genotype samples per year and 200,000 genome profiles per year. These are kind of rough targets over the next 4 to 5 years for the broad community of crops that we're supporting, and to try and hit some of the volume targets that we want to reach in order to drive prices down. I'll just spend a minute here talking about kind of, this is an exercise I did a few years back with the Maze team on kind of mapping out a decision tree, and just the complexity of it, to highlight the complexity of how a person might think about different strategies in breeding. And the point here is really just to make that, I think Michael mentioned it earlier, there's a lot of decisions that a person can make, and this is just in the space of, you know, how you might apply markers, yes or no, which way you might think about doing it. Obviously, there's a lot of other decisions that go on in parallel or at the same time. The point I just wanted to highlight here was that one of the things we're trying to do in working with our Module 5 counterparts on sort of decision support tools to enable efficient ways of thinking about managing your breeding program to make the most of opportunities that are there. So I just use this as an example of, you know, this is kind of a mock-up of a population selection tool that you could envision. In this case, you know, we have a couple traits so that we're interested in. We've got some elite lines and some trait donors. You might think of them as, in this case, the example I'm using, they might have high zinc for a breeding program. And then, you know, a breeder would be able to look kind of at the trait values of those potential crosses and see where the opportunities and where the weaknesses are for different traits. And then, you know, we may also be able to look at, you know, our historic germplasm and see how well do we think we could predict this given population, given our information. That would give a breeder some indication of whether GS is a good strategy, or whether we just need to phenotype these in order to, we don't have a good training set for this particular population. And then also some information about which markers might be segregating in each of these populations to see where there are opportunities. So I just mentioned this briefly because I think the decision support space is as important as anything in terms of really making this deployable and usable by the breeding programs. Okay, so our intended result is to see us, you know, focusing our resources on developing and executing implementation plans on Greenlight, MAS, and GS applications at lower cost. That's what we're trying to enable. Yeah, and our milestones, we have a few kind of high-level milestones. We'd like to see our breeding parents routinely characterized with high-density profiles that will enable us long-term to see the opportunities for forward breeding and GS, and then implementing workflows for the target traits within the product profile context. And finally, yeah, QC used at strategic places within the breeding pipeline. So that's a lot of what we'll be looking at in Module 3. So we really hope this week that we have opportunities to dialogue with the breeding teams here at IATA and your partners who are present. Try to understand what the needs and constraints are in this whole area of where genotyping applications could benefit your programs. have good discussions about strategy and framework on the cost-benefit analysis. We want to start those discussions, recognizing that that will take some time to develop, and but at least start to get some of that information in our minds, and then really strengthening the relationships. And I have to admit that you know, even this morning I'm seeing so many familiar faces, and you know, I interact with people so periodically and in different places I can never quite remember. So my apologies up front at the beginning of the week. If I know you and I've met you and I don't remember your name, my sincere apology, but I look forward to, by the end of the week, walking away with stronger connections with many of you who will be working with EIB Module 3 in the future. So with that, thank you.